Good day. We carry on with genetic notes. This is the fourth page, so this is part 4, and this will be our last part of it. So we've done that, we've done that, we've done that. And we arrive at things called genotypes and phenotypes, and eventually we'll get to Punnett squares. Paired symbols show the genotype of an organism. So geno, you look at that and you say, oh, the genes, these are hidden away, things we can't see. But we know that they're related now to the DNA. So genes are sections of the DNA, and we're worried about specific genes. So a genotype there, we have a combination, in this case, of 2 tall genes. The effect caused by the genotype is called the phenotype. Pheno means to show. It's what's expressed. It's what we can physically see. So big T is going to be tall, and we call it homozygous dominant. Big T, little t is tall, and we say heterozygous. So this is what we can see, the genotypes hidden away. Little t will be short, and it's homozygous recessive. So here we have 3 genotypes, but there are only 2 possible outcomes, so we only get 2 possible phenotypes. The genes that have contrasting effects in a trait are called alleles. So alleles, in a sense, is just another word for gene, and often we'll use the word allele or gene to mean very similar things. We will get into more exotic genetics, but we're going to start with simple stuff. Now, Punnett squares and probability. These show us the possible gene pairings from a cross. So that means when we have 2 different organisms contributing sex cells, what kind of combinations can we get? There are some times where it's 100% guaranteed that we'll get something or won't get something. And that's if there's a 100% chance or a 0% chance of it happening. But if it's not 100% or 0%, then we have to worry about probability, the odds. What could we get? So we look at this and we say, hey, we have big T, and little t, little t. So we said, here's our big T, and here is our little t, So I'm going to attempt to type in here. Let's see if it'll cooperate. So I get big T there, and I get little t there, and we'll make those red. Come on. There we go. And then we do the same thing down here. So I went big T, gene pairing. So then I went little t, then I went big T, in this box, little t there. So this is called a Punnett square. And from this cross, we fill in the 4 boxes, and then we interpret what these things mean. We see there's no chance of getting 2 big T's. But in terms of our genotypes, we can get a 50-50, or 2 out of 4 boxes are big T, which is heterozygous, and that means a 50% chance of tall. And then we can see we have a 50% chance of short, and that's what it's saying here. So this is the Punnett square, and then this is the interpretation of Punnett square. Now we have some more examples to try down here. So I guess I better do some typing, because these will be examples. You can try them out, and then you can check against my answers in just a moment, and we'll make that another video.